Welcome to the latest edition of the Mind-Gut Conversation podcast, a place to learn about new ideas from thought leaders in the area of optimal health, the science of mind-gut microbiome interactions, food, and the environment. Today, I have the great pleasure to speak to Joel Dore, an internationally known expert on the gut microbiome. Dr. Doré is research director at the French National Research Institute for Agriculture, Food, and the Environment. He's also the scientific director of Metagenopolis, a platform dedicated to quantitative and functional characterization of the microbial genome, funded by the French government Futures Investments. Joel Dore received his PhD from the University of Illinois at Urbana-Champaign in the US in 1988. His main research interest is the molecular assessment of the human intestinal microbiota in health and disease, and investigation of the molecular crosstalk between intestinal bacteria and human cells. Dr. Doré has published more than 220 publications in peer-reviewed scientific journals. His goal is to provide a better understanding of the intestinal ecosystem in order to support therapeutic choices in the medical area, as well as health claims for functional foods. Welcome to the show, Joel. So let me start out with a sort of a general question that's often asked by a lot of people that are not necessarily experts in microbiome science, but it has to do with the utility of currently available stool microbiome tests. So there's several tests available based on the technologies, the majority, the 16S sequencing, then shotgun metagenomics, and transcriptomics. So first of all, is there a big difference in terms of the quality of the test? And secondly, what is that difference, and what do you think is the state of the art to do it?
It's really a matter of debate. Here in Europe, we have several companies who do offer microbiome tests. Results usually take the form of a long illustrated report, with lots of nice figures. And our medical colleagues tell us this is useless for them. And tribunes in newspapers and from professional societies also do insist on the uselessness for medicine.
Mhm.
And I personally have, let's say, a more open view on this, as we need structured pipelines in the hands of medical biology if we want to see an advance towards medical use. And sometimes the critics goes as far as saying that this is charlatanism and there is no need to even consider, or you'd think that we will never have what we need to have to make some progress. And so that's why, importantly, I do see two possibilities or two possible demands. One for wellness, or sometimes you hear of holistic care, or just curiosity, as we were curious about our genomes when genome sequencing became available. Why not our metagenome? And two, medical use as an integral part of the decision process. And so for medical use, it has to tick a series of boxes, as I like to say, starting with the standards. You want the same sample analyzed in 3 places to give the same result. And this is very important. It has to be confronted to large reference data sets. And this is upcoming. There are many large general cohorts being structured today. It has to, well, we need the structured analytical pipeline, as I mentioned, in the hands of medical biology to move towards the proof of clinical benefit in patient management. And this will also require some regulatory aspects. And that was actually what the recent US publication complained most about, saying that FDA should state whether it is wellness or whether it is medical device in vitro diagnostic. And well, I think this is indeed, if we want it to be of medical use, then we need to have the regulatory path, obviously. And then at some point, there will also be need for training of healthcare professionals for them to be familiar with what comes out of it. So here we come to your question on technicalities. You mentioned 16S. So this is a single gene approach, one gene reporter for phylogenetics of microorganisms. It only usually talks of bacteria, and it relies on processes that, whenever we tested, do not seem to be standardized and even standardizable. The second you mentioned is shotgun metagenomics, so still addressing DNA, but all possible genes. And we published actually that in 2017, showing that, yeah, it can be standardized. So it could be applicable for the medical application part that we mentioned. You also suggested transcriptomics, I think. Transcriptomics relies on RNA. It's more for expression. There is a question whether it's reliable or of any use for the fecal material that comes out after at least 20 hours of transit in the gut. So we still don't know whether this would be informative at all compared to what happens inside the gut. So it's question, and it's still not mature, I would say, technically speaking. So shotgun metagenomics for medical application, I think this is ready. And we have this, Le French Gut, we call it, project in France, where we will recruit 100,000 people. We are getting near to 15,000 now. And obviously, this is one absolute need to have the reference to be able to consolidate evidence of signatures, for example.
So do you think, so before we go on, so do you think, based on what you said, is right now, if somebody gets one of these stool tests, and I mean, I see a lot of patients in my practice that come with these 15-page reports and have no idea how to interpret it, and their doctor couldn't do it either. And is this a waste of money right now if they say, you know, I have, well, I mean, people come with IBS or people come with other chronic abdominal pain symptoms. Is this a waste of money right now to do that test?
I have read many, and I think that I can exploit this information, but after 40 years of work on the microbiome. And so I think that in the current situation, it's a bit tricky to really exploit information. And there is also this situation where people come with a report with 40 pages and sometimes recommendations. And the recommendations that I see occasionally go far beyond what science would allow us to say. So I think that there is a bit of exploitation of credibility of the people on that matter. So I think it's not really too fair at the moment. What I envision in terms of what will be the future for medical application is it will not be a report of 40 pages with lots of figures. It will be maybe one line or a few lines, like the line of cholesterol in your biology report, saying, well, you are there. The general population distributes as such. And in your case, microbiome is not concerned, or microbiome is for sure concerned, or you're in the gray zone. So we will do something with your nutrition, for example, try to see whether we can twist it towards more diversity, for example. And let's meet again in 3 months and see whether we have managed to move a little bit this one parameter. But I don't foresee usefulness of large fancy reports for the moment.
And recommendations, I mean, this is, you know, you mentioned it briefly. I mean, recommendations for specific dietary changes. So I've seen a report from a friend of mine that said, you know, you should not eat blueberries. I mean, this is kind of based on this is complete fantasy.
Well, there is no science that would support that in any way. So for sure, we are still on really crude basics, I would say. So if there were any recommendations, it would be rather general, like well, more fiber would probably help, for example. Modulators of the microbiota at the level of individual food items, I don't see that coming for the near future, for sure.
Okay, thank you. So yeah, so the third of these questions was, are these tests or any of these tests useful to predict disease risk for like chronic diseases like depression, neurodegenerative?
Yeah, so we are not there yet. Recurrent features in chronic conditions post onset, so for patients, include often low microbiome richness, I mentioned already, and also loss of certain dominant symbionts, especially those that can be involved in the production of specific molecules like butyrate. Butyrate is a short chain fatty acid, and it's involved in a crosstalk with human cells. It's an energy source for our epithelium. And it's also interacting with the immune system. But this is association. It's not prediction of risk. And the only exception in terms of prediction of a chronic disease is the context of inflammatory bowel disease. And it's coming from a Canadian work with a GEM study where they followed people a long time, actually, after sampling. And they went back to analysis of the samples for new onset patients. And the same parameter actually were predictive when we mentioned loss of richness and loss of certain symbionts. So that might in turn be predictive of altered calprotectin levels. So the beginning of inflammation as much as 2 years before. So this is one specific case telling us, yeah, it's very possible in the future that we are able to identify such predictors for other chronic conditions. But it takes longitudinal studies and the follow-up of patients in terms of clinics to be able to do that. So there aren't very many pieces of work that actually address that still today. What is well established, though, is that low microbiome richness by itself will predict non-response to a dietary intervention in overweight and obesity, for example. This is work I contributed to. We published in 2013. And also, it's predictive of a more rapid aggravation in liver conditions. You know, when people go from steatosis to NASH to cirrhosis to extreme situations of cirrhosis where there is what is called ACLF, acute on chronic liver failure. Well, low richness microbiome predicts that you will reach this level faster. And in cancer therapy, low microbiome richness can predict lower progression-free survival upon therapy or more frequent complications such as graft versus host disease, for example, in blood cancer therapy. And so these cases of prediction are also quite meaningful. If we were able to provide material or a test for the clinician, then that would make sense for the clinician to integrate this information in its decision process. And we have colleagues, for example, in France at Institut Gustave Roussy, who work on cancer and who actually now are developing a systematic collection of fecal material from patients who come for immunotherapy, because they know that the picture of the microbiome will tell them whether the patient will be among the 50% responders or 50% non-responders to the treatment. And if non-responder, then there starts to be ideas of what should be done before. So I'm expecting that we will see a progress along those lines in cancer therapy in the near future.
Yeah, it's interesting that of all the chronic diseases, cancer, you know, which you would not have thought a few years ago before these first studies came out, would be the
For
one
sure.
that, yeah.
Actually, cancer therapy, we're not in predicting onset of cancer yet. We are happy. It's quite amazing, and it's made progress very rapidly, as you are suggesting. Yeah.
You mentioned, I mean, going on to a next sort of clinical question, is there solid evidence to say that the gut microbial diversity and richness plays a role in resilience to several diseases? So this is not about therapy, but this is actually about being, you know, resilient to, for example, like
Yeah.
COVID-19, or I recently heard a talk here at UCLA, actually a couple of days ago, for HIV infections, that the microbiome, so the pre-infection compositional microbiome had a certain value in predicting the the likelihood that somebody would develop HIV.
Yeah, baseline microbiome
Yeah.
is quite important actually in many contexts. As I mentioned, we've seen that the richness of the microbiome is a strong parameter, and we also combine that with the ecological composition or ecological structure of the microbiome. And in obesity or cardiometabolism in general, when you have a dominance of certain groups of bacteria, so a specific ecology that is called Bacteroides dominated, and low diversity, then it's really the worst microbiome configuration. It's one highly prevalent in rapidly evolving or more severe patients. When we analyze the distribution of richness among healthy individuals, we usually have a small fraction of population with low richness. It's around 10% or so. When we analyze that in moderate obese or even overweight individuals, then it goes up to 25% or so. If we do that with extreme obese individuals with BMIs of 40, 42, then it's 75% of this population with low richness microbiome. But then I would talk of resilience to stress, but rather resistance to disease, let's say. Microbiota richness is predictive of many conditions, as we mentioned. Rich microbiome, is protecting or preventing so much that it would be what I would consider aiming for a rich microbiome as a relevant measure of prevention overall, let's say. So when I do think of I can use nutrition to prop my microbiome, then the measures I take is going to directly favor richness, fiber, and diversity of fibers. We can discuss that further if you want.
What's the link between the microbes and these disease risks? Is it the immune system that a lower richness basically has an effect on the immune system, and that's the mechanism in the human body then that.
Yeah. So strong point, very important point. Actually, I spent most of my career working on the microbiome, thinking that it was the end of the story or the core of the story. And nowadays, I talk a lot of symbiosis, of host microbe symbiosis. And I think that this is really the key. Microbiome itself will do nothing, but just if you consider the interaction between the microbiome and the host, when it comes to the gut, it's interacting with the gut wall, so it's linked with permeability. When both alter, actually, you will tend to have a rise in inflammatory tone because of inflammation coming, because of signals going through the gut wall. And when there is inflammation, there is oxidative stress. And just those four parameters, the microbiome, permeability, inflammation, oxidative stress, can really turn into a vicious circle. This means that the system may be resistant and resilient up to a certain point. You can call it a tipping point, actually, beyond which it may dramatically change in configuration to enter an alternative state that will be sustained by the vicious circle. And it is known by complex system theory, actually, that if this happens, then the return to initial configuration can be very complicated. And I would not exclude that chronic conditions we are facing, those we've seen rising in incidence for now 3 generations or so, uncontrolled, may be situations where this dysbiosis is just not a story of the microbes, but one element of a system that is altered by many facets, as we just illustrated. So it's not just a microbiome story; it's an altered host microbiome story.
Yeah, I find that this concept really intriguing. It's kind of a holistic science, more or less. I mean that you can that you don't look at, I mean, particularly you don't look at individual microbes, you look at these interactions, systems, biological interactions. And I think we're just beginning in science really to accept that concept.
Yeah, it's a challenge intellectually speaking, because we like to narrow down and to focus on functions and things like that, or pathways, or even single elements of a pathway. But then what this tells us is we have to look broadly we can explain that a bit further for diagnostics, because just looking at the microbes is not the whole picture. So we should be combining elements that tell us about the state of the microbiome, but also about the states of the gut wall, inflammation, and oxidative stress at the same time, just for these 4 parameters. It's probably a simplistic view still, but having more than just the microbiome seems to me very important for diagnostics or for prediction, but also for treatment. We've done a piece of work recently where we use the model, you'll be interested with that, the model that is used to test new antidepressant drugs. And so it's a model of mice that you stress for 4 weeks, and then they're established in a situation where behavior-wise, you can measure their anxiety and their depression, and you take them 3 weeks later and it's still there. And if you treat them with an antidepressant, then you may completely correct the alteration of behavior. And we speculated that because there is measurable inflammation and measurable alteration of the microbiome, that possibly our vicious circle was established. And so we proposed to treat them with 3 bioactives. One, glutamine, that will essentially nourish the gut wall. Another, curcumine, that is a polyphenol that will essentially act as antioxidant and anti-inflammatory. And then we added a probiotic, Lactobacillus rhamnosus GG, which is known to act in many aspects. It's recommended by the gastroenterologist in association with antibiotics, as it increases resistance and resilience of the microbiome. But it's also known to protect gut permeability, to act as anti-inflammatory, and it has a catalase that is a matter of antioxidant. And so, well, we used the 3. We used them separately, 1 by 1, 2 by 2, and 3 together. And to make it short, when we have the 3 together, we do as well as clomipramine to restore normal behavior in our mice. Clomipramine, which is an antidepressant drug that is commonly used in France, actually, that in humans has secondary effects very often. Well, here we are using 3 food-grade bioactives, basically, and we do a very impressive effect on correcting behavior of mice. And so what we did, we started a human trial not too long ago in patients, so we cannot we have to use the treatment, the normal treatment. So we selected patients that are escaping from the first line antidepressant drug and that go onto venlafaxine, and they receive or not, it's a randomized control trial, the combination of bioactives, or 3 bioactives. And we're, well, this is ongoing. We will know in 2025, I think, because it's 92 patients to be included in the process. But yeah, we're quite optimistic. But basically, with respect to what we were saying, the concept of symbiosis, of acting on the microbiome and host parameters, seem to make sense, actually, in this case, where you can correct a situation where you have a combined alteration of the microbiome host parameters if you actually try to tackle parameters. And well, possibly this would apply to other chronic conditions. In liver conditions, for example, you know, it's a continuum between steatosis and NASH and cirrhosis. And at some point, the clinicians tell us that it's irreversible. But at some point, it's irreversible. It means that if you can prevent from reaching this point, then you can probably go back to normal. And possibly even at this point, if you act on all parameters and not just inflammation, which is the usual symptoms that are treated, then it might be possible to be more efficient. So I view the possibility of acting on host microbe symbiosis as a sort of an add-on to currently applied therapies.
Yeah, I mean, coming back to your depression study, I mean, I find that fascinating because it really supports this new concept of nutritional psychiatry, where
Yes.
which has become very, I mean, I wouldn't say very popular, but certainly there's now a few individuals who promote that message, that milder forms of depression, or yeah, I don't know how I would sort of look at it. I mean, look forward to see a study in human subjects, but the main test is going to be, as a psychiatrist, if somebody comes to you with severe depression, would you start out with these three components targeted at the microbiome rather than starting with cognitive behavioral therapy and an antidepressant? But maybe physicians or psychiatrists won't have to make that choice. It could be a combination. You could.
I think it should be, actually. I think that in diabetes, for example, the obvious nutri-dietetic recommendation is here. Well, maybe it should also be here in many other chronic conditions. I would say especially if there are intestinal symptoms, because one of the obvious sign of something being wrong is that you have altered transit time or pain, intestinal pain. And if this is the case, then you're not dealing with something that is only concerning your brain, actually, your mental health, but also your physiology, your gut physiology, and acting at the same time as your usual standard of care on these parameters might be increasing chances of being more effective.
Yeah, let's go from the psychiatry topic to something that we're also experiencing, and progressive epidemic, the obesity epidemic, and I've sort of had an interesting experience with a few social media posts about these new drugs, the GLP-1 agonist and GIP agonist, and some of the comments that I got to my post was, we have to deal with this epidemic with lifestyle modifications. Well, I mean, the reality is, that hasn't worked. I mean, people have tried for all kinds of diets and behavioral modifications of diets, and we still have a progressive increase of the prevalence rates of obesity. So what role do you think the microbiome has in that epidemic?
So obesity is really complex. It's very difficult for me to consider we could simplify the message. So I want to start with that. My understanding is that there are heterogeneous configurations, likely obese yet healthy individuals, some that can run marathons, as I understand, for which the microbiome may be playing no role at all. And as well, there are obese individuals for which the microbiome is probably a causal factor from onset to maintenance to aggravation of the context. And in such cases, I would expect to see a combination of disease and intestinal symptoms, actually, that could justify to address gut symptoms as an add-on to our regular treatment, as we were, probably other chronic conditions. In obesity, what is well documented is when it comes to microbiome, you have dysbiosis very often rise in gram negatives. You have LPS, so endotoxemia, and you have migration of LPS in the tissues, and that attracts macrophages and so on. You have inflammation in the tissues, and that will pollute the good functioning of insulin sensitivity. I mean, I don't think that this is the general situation, but when the microbiome is at play, then it should be addressed at the same time as any other element you want to go. You mentioned GLP-1 agonists and so on. I mean, I don't know that very well. It's not my field, and I'm not a clinician. But in fact, when I read that they could actually be acting as anti-inflammatory,
Yeah.
well, for me, that means that they should actually, just on that basis, restore a little bit of the potentially altered symbiosis between the host and the microbes. And so, but then you would want at the same time to have um, really marked recommendations on the diet, because if you have a sustained, completely altered dietary behavior, then you have all the elements you need to maintain the vicious circle and to maintain an altered context. So it will be very tricky to have an effect, especially if you have this element of what is called a critical transition. You know, you bounce from one state of equilibrium that can be health to one state of equilibrium again, but that can be pre-disease or disease. And this is when the return may be very complicated. So if you want to really return, then you have to pull every string that you can find. And yeah, acting on the microbiome can be one. If your best drug will also act indirectly on the microbiome or on the symbiosis, then it's all too perfect, actually. So
Yeah, this
yeah,
is something. I mean, you've mentioned this earlier in the conversation, and I've always been fascinated listening to your talks about this, the systems biological view and um, the critical transitions, and this is almost something I think that is at the core of these chronic diseases, because like if you look
yeah.
at obesity, people have done extreme things like gastric bypass surgeries. I mean, also with these new drugs, the minute that people stop them, they regain their weight. So
Most likely, when actually for those for whom it might be a question of vicious circle established, then it's not sufficient to have this one drug acting on one specific pathway element to do the return. We need to work more on that, because the example I like to give, and it's a bit extreme, but in nature, in environment, and also in economics, vicious circle and critical transitions do apply. And in nature, one of the examples is the lake that turns green because of a rise in algae, and they use up oxygen, and it messes up the system basically, because you don't have the oxygen to support the life of their normal predators. And Martin Schaeffer, who in Holland works on those model systems of economics, said, well, if your lake is green, then remove the fish, because they are the ones that are eating the few predators of the yeast that are remaining, and that should restore the system, or not the yeast, but the algae. And he did experimentally the demonstration. You remove the fish, then the crustaceans thrive, they eat up the algae, and the lake returns clear. And well, I wish we had an option like that for the human gut, where our best option would be to have the possibility to do ecological knockouts. If we could say, well, I don't want to have this species or this phylum or whatever, and then I use this molecular trick, it's gone, and I see whether my vicious circle is back to normal or not. But I think that if we include that consideration in the way we think, then we may actually be progressing. And I'm not thinking of revolutionizing the system at all. It's just a matter that in medicine, for the moment, people deal with symptoms. And in many of the chronic conditions, the only symptom that is sufficiently visible is inflammation. So people deal with anti-inflammatory drugs. with IBDs, it's anti-inflammatory, it's immunosuppressive, it's biologics. And if it doesn't work, you have to remove a segment of the gut, actually. And I think that this is short. There should be a combination of action on inflammation, on the gut wall in terms of repair, and on the microbiome as well. And possibly, why not, on oxidative stress in the system. And so, yeah, I think we have to, it's complicated, but we have to open to more possibilities of combinatorial approaches.
It's sort of interesting that, I mean, this kind of new thinking, this new systems biological thinking is really something that's happened in environmental sciences. I mean, people that talk about climate change, it's very similar. You know, there's
Yes.
not.
Yeah. You talk of climate change. I mean, there are many vicious circles that we are suffering from. We'll see, ice caps going.
Yeah.
Well, they actually reflect sunlight, so they limit warming, but warming melts ice. So actually, there is less refraction or reflection of the sunlight, and so more warming gives to more melting ice, and so we will lose Greenland for sure. And this is the one vicious circle at play.
Let me come to a sort of a general mechanistic question also. So the main question is, are the unique characteristics of the gut microbiome adaptations to a rapidly changing environment, or what's called the exposome, diet, medications, environmental chemicals, forever chemicals, microplastics? I mean, it's a lot of growing list of these environmental factors. Are these changing environmental factors causing a mismatch between a very slowly changing innate immune system or our gut properties, resulting in low-grade immune activation? In other words, so the microbes adapt quickly, probably within days to a new environment, but our gut is sort of dependent on its genes, and the genes obviously don't change that fast, thousands of years. Is this mismatch really one of the main factors for this chronic low-grade immune activation?
I think this is probably at play, but I would think more in terms of across generations or through several generations. What I mean by that is that, the microbiome will evolve fast, will respond fast to the environment, but it's overall for one person adapted to its immune system, and its response is usually, well, I don't have my ecological favorable system anymore, so I calm down, but I'm there. I just mellow down. I go 2, 3, 4 logs down, so I'm subdominant. And then when things go back to normal, then I can actually return. This is what happens when we take an antibiotic. We mess up completely the dominant microbiota, but then when we remove it, it comes back to almost exactly what it was before. Unless you do it chronically every month or so, or for a long period, you actually we do see resilience. And so for one generation, I'm not sure that this would be at play. But then, I mean, I think that among the tentative explanation of the rise in incidence of the chronic conditions that we have seen, rising uncontrolled, as we mentioned, for me, 3 factors: change in nutrition, change in everything that surrounds birth, that will matter early on in life in terms of establishment of the relationship we build with our microbes, especially the microbe immunity relationship, and then exposure to environmental factors, those that you were mentioning, chemicals, plastics, many xenobiotics that our bodily cells have not been able to cope with because they evolve very slowly, obviously, in terms of genome evolution. But there are microbes can do nothing but try to cope with. And so over 1 and 2 and 3 generations, then actually we will accumulate losses in a way. This is what we do when we compare microbiome of Occidental population with microbiome of tribes that have remained away from modernity up until now, basically. We see that we have lost sometimes 50% of the richness of the microbiome, the dominant microbiome. And so this is a cumulative effect over time. And I think that this is leading to a lesser resilience for sure. So the system will have a tendency to reach the tipping point faster. And so a larger fraction of the population may actually bounce into chronic conditions faster. Some are really scary. I mean, you know that very well, better than I. But for autism, when I look at the rising incidence, it's really concerning. And so you'd want to be able to do something about it as soon as possible.
Yeah, I mean, I mentioned briefly, the microplastics. So there have been these recent publications, really shocking, that even in bottled water, that microplastics are also being discovered now in the body, in coronary arteries. I mean, this is something that's pretty very slowly changing, only because I'm always surprised that, like, on airplanes or in hotels, still plastic bottles are the predominant way of distributing water. If this is really something, I mean, we have no idea. It's kind of a black box. What do the microbes do to these microplastic particles or nano plastic particles? And what's the consequence of that for our gut, for our health?
Yeah, for microplastics, I really don't know. I mean, the best documented segment on the chemical exposure for me is that of Benoît Chassin, who was in Georgia for his postdoc, and he's now in France. And he worked on additives on emulsifiers, and showed that emulsifiers act on the microbiome and they act on the gut wall at the same time. So you have a combination of permeability and dysbiosis. And of course, this is not a good cocktail. And it will promote a higher risk of development of gut inflammation, higher risk of development of obesity if you challenge it in animal lab animals, for example. So it's a nice demonstration, but in this case specifically for food additives, what Benoît was demonstrating is that if you consider 50 of them, and there are many that we have or are exposed to every day, if you take 50 of them and you rank them in terms of severity of their effect, well, you have a very marked ranking, actually. Some are very detrimental and some are quite normal, I would say. And so if the industry, if our regulators were to account for that, then we could do better. Mm-hmm. We could swap some for others and have ice cream that would not be so bad as they are today.
Yeah, no. So, yeah, I think we've pretty much come to the end of our time here. I mean, I could talk to you for hours, particularly.
Same here. Sure.
Particularly because I sort of really resonate philosophically with this systems biology concept. I think this is something science, in all aspects of science and even society, will move towards, to understand our world in a different way, not in this linear, reductionistic way, but in seeing the whole. And to have somebody like yourself, one of the most prominent microbiome scientists, to embrace that and use this in your presentations, I think for me it's always really exciting to listen to that.
Thank you very much. Thank you, Iman.
Okay, Joel, thanks so much for the time.